AP Biology Genetics Part 5. Today we're going to start off with a practice problem combining your ability to find the genotype of the parents by analyzing the offspring and doing the chi-square test with with 4 categories in the offspring. And we'll see if you know how to do this. So let's go ahead and pause the podcast at this point and copy down this problem. Here we have big P, little p, that's representing purple and white as before, big T, little t. And the parent generations can be purple and tall, crossed with white and short. And the F1 are going to have the following ratios: 210 purple tall, 170 white tall, 190 purple short, and 205 white short. So what you're going to try to do is figure out the parent's genotype and find out if the chi-square test supports your hypothesis. So go ahead and pause now and start thinking about what you're going to do to try to solve this problem. Alright, I'm going to go through the answer step by step, and at any time the answer is unclear, make sure you ask those questions in class. The first step is to look for the children's characteristics that can help us understand what the parents gave them. So if we take a look here, we have for the first trait, and remember we analyzed the traits separately, we have 400 purple and 375 white. So it looks like a 1-to-1 ratio of purple to white, and that's going to be useful for trying to figure out, what the 4-square Punnett score is going to look like for that first trait. We also have 380 tall and 395 short. Also looks like a 1-to-1 ratio. So remember, analyze traits separately is your first step. Pause at this time and think about the next step. All right. The next step is, what do the parents have to be to make 1/2 purple and 1/2 white? So we'll just analyze that first trait first. So the first parent is purple and tall. The second parent's white and short. So purple, there's 2 ways to make that. You can either make it with big P, little p, or big P, big P. White is only one way, little p, little p. So the only question is, what is that purple? So if the parent is big P, little p, crossed with little p, this is our Punnett square. If the parent is big P, crossed with little p, we get all purple. Okay, so if we do the Punnett square for a heterozygote, big P, crossed with little p, we find that we get half are going to be purple and half are going to be white. And that's exactly what we find in the children. So we should, at this point, suspect that the first parent's genotype for the first trait is big P, little p. Now for the second one, what are the parents to make 1/2 tall and 1/2 short? Well, it could either be the second parent's going to be little t, it's short, so there's only one genotype possible for that. If that first parent is big T for tall, then they're all going to be tall, and that's not what we see as far as the offspring. What if the first parent for the second trait is big T, crossed with little t? Well, if you do that Punnett square, you find you get half tall and half short, and that's what we find in our offspring, about half tall and half short. So now we got to do the chi-square test. We got to find out if our results, if the offspring are close enough to the expected ratio in order to draw the conclusion that the parents are probably big P, little t, crossed with little p, little t. At this time, pause the video and try to figure out how you're going to do that chi-square test. All right. So practice continue. We have our chi-square test. The first thing we got to do here is find the expected. So let's count up the number of offspring, the F1 generation. If we count them all up, we get a total of 775. So what percent of that what number of that 775 are going to be purple? What number are going to be purple tall, purple short, etc., etc.? So to do that, we have to use our math method of figuring out these kind of problems. If you do the first trait first, big P, crossed with little p, we get half purple, half white. If we do that same thing for the second trait, we get big T, little t. Half are going to be tall and half are going to be short. So to find out purple tall phenotype, that's a half times a half, gives us a quarter. 1/4 of 775 gives us a total of 194 were expected to be purple and tall. And if you do that for all of them, you find out that every one of the categories, we should expect about a quarter of that group, the F1, to exhibit those traits. Purple short, about 194. White tall, expect 194. White short, expect 194. So that's the next step in solving these kind of problems for the chi-square. So then after that, we got to fill in our table. Now, if you remember, this is the part that's going to require some memory. It's observed first, expected, observed minus expected, then you square it, and then you divide by expected. I don't know if you can come up with a mnemonic trick for that. OE2E, maybe, OE2E. Observed, expected, observed minus expected, squared, that's the 2, and then over E. If you can think of something better, let me know. All right, so for purple tall, we have observed 210, 190, 170, 205. That goes in the first column for our chi-squared, figuring out chi-square. Expected, 194 for each one of them. If you subtract observed minus expected, you get 16 for the first category, minus 4 for the second, minus 24, and 11. Then you square it, you get 256, 16, 576, 121. And then you divide by expected, and you get these following values. If you add them all up, the sum, which is going to be your chi-square value, is 4.99. So moving right along, we have our chi-square value. The sum of all those numbers combined is 4.99. Now remember, degrees of freedom, when we look at our table, is going to be the number of categories minus 1. So if you take a look here, we have 4 categories. Minus 1 would be 3. We have degrees of freedom of 3. So now we've got to compare this value to our table. All right, and you don't have to memorize the table. They'll always give that to you on a test. Chi-square is actually represented by this little symbol here. Looks like a little X with a little square on it, so I'll just kind of put a little thing there. Remember, you have to find the value in the 0.05 column because there has to be less than a 5% chance that your results are due to chance or some other factor. And if we have degrees of freedom of 3, listed over here, we'll just look straight across, and we get a value of 7.82. Since 4.99 is less than 7.82, then we can accept our results as being valid. So these are probably the parents' genotypes. Big P little p crossed with little p little p. Sorry, big P little p, big T little t, crossed with little p little p, little t little t. Results are valid statistically. These are the kind of tests that you would have to do in order to publish your work if you were a researcher. And this is kind of like your introduction to that kind of stuff. All right, for the rest of the lesson, we're going to cover some other topics in genetics. Queen Victoria and her descendants. This was her family tree, and if you notice, one of the things that kind of stands out is all the males are getting the disease. Now, if you remember from the previous lesson, I think it was the 4th part of genetics, that usually males get a sex-linked disorder, and hemophilia is a sex-linked disorder. So the women tend to be carriers, the men tend to actually have the disease, and that's one thing to look for if you're trying to figure out what kind of disease it is, looking at a pedigree. All right, X inactivation. Female mammals inherit 2 X chromosomes, but only one is active during the adult life of the female. So the other one, and this is randomly assigned, becomes inactivated. And this is called a bar body. An inactivated X chromosome is called a bar body. Now don't confuse that with a polar body. A polar body is one of the 3 cells that have been made during meiosis by females. One becomes an egg, one of the 4 cells becomes an egg, and the other 3 are polar bodies that just get reabsorbed. This is something else. A bar body is the X chromosome that gets inactivated. And sometimes we hit some interesting patterns on cats as a result of this. If you take a look at this cat, the gene for fur color is carried on an X chromosome. So if you inactivate the X chromosome with the black fur allele, then the cells that divide from this cell will all be brown, and that causes a brown patch of fur. All came from that single cell during embryonic development. Or if we inactivate the brown allele that's on the X chromosome, then all the cells that are produced from this cell will have black fur, and that gives us some interesting color patterns. This is also seen in tortoiseshell cats. They have the same thing going on. Whether they have orange fur or black fur activated, depending on which X chromosome is inactivated, will give them these color fur patterns. Now, a male cat's not going to do this because they don't have 2 X chromosomes. So males will never, in the cat world, have tortoiseshell colored. Don't get those confused. Bar body, inactivated X chromosome, and polar body, 3 of the 4 cells produced by a female after meiosis that will not form an egg. Male pattern baldness, just a quick lesson on this. In males, the baldness gene is a dominant trait. So if you even get one of them, no matter if you have the normal allele, you're going to go bald. However, for females, you have to get 2 of the alleles, and in females, it's recessive. So in males, it's dominant, females recessive. So females have to get 2 of those alleles, one from mom, one from dad, in order for them to go bald. So women can go bald too, but it's a lot more difficult. Kind of like with the sex-linked disorders of hemophilia and muscular dystrophy, which is a different concept, X chromosome disorder or sex-linked disorder. But Another reason why women tend to get less like baldness and other diseases is because of those X chromosomes, and in this case, recessive traits in females versus dominant traits in males. All right, so mechanisms of inheritance. Let's skip this for now. We have mechanisms. Okay, so the basic idea here is that DNA is coding for these proteins that give us our trait, and these proteins will actually make the trait. They're kind of like the cellular machinery, and then the trait is made. So you mess with the DNA, you mess with the trait, and that's kind of what we're going to be talking about after second quarter. All right, so dominance. Just because something is dominant doesn't mean it's better or more common. For example, if you have 5 fingers and 5 toes, you have the recessive alleles. You have, like, let's say little p, little p. If you get big P, you got something called polydactyly, and that's a dominant allele. That means you're going to have extra fingers and such. So just because something's dominant doesn't mean it's more common. And this is one example of a dominant allele, a dominant trait, when that's not very common in populations. Here we have more polydactyly. Usually they just cut them off. And it happens in one about 400 people. Most people are little a, 5 fingers, 5 toes. There's a famous guy who had, he was a singer or a guitarist, and he was one of the better known people with polydactyly. All right, so here's some genetic diseases that are recessive. That means that if you have the normal allele, you're going to be okay, but you might be a carrier. If you have 2 of the normal alleles, you'll be, let's say, big C or something, and no problems. The only problems that happen in the children are when you get 2 of the recessive alleles. So let's talk about that now. So why does this happen? Well, typically people that have cystic fibrosis, let's say it's little a, are not going to have kids. They're not going to want to pass that on. However, carriers will not know they have the recessive allele. Let's say this is for cystic fibrosis or any other recessive disorder. 2 carriers fall in love. They have kids. Well, 3/4 of the time, the kids are okay. However, 2 out of 4, half the time, you're going to produce carriers. 1/4 of the time, however, the kid's going to get the recessive allele from both parents, and that's going to result in a kid with the disease. In this case, little a, and this could represent like cystic fibrosis or Tay-Sachs or anything that's a recessive disorder. The reason why these disorders don't go away is because of the carriers. So if you knew that you had this in your family, if you got married, you'd want to make sure that your spouse had a check, genetic test to make sure that you weren't going to have kids that have the disease. Tay-Sachs is a disease that affects mainly Jews of Eastern European descent. Now this is just where the ethnic group first had the mutation to the gene for the lipids, or the enzyme that breaks down the lipids. It has nothing to do with religion. As far as what happens, well, it strikes in about 1 in 3,600 births, and basically it's the lysosomes, the enzymes in the lysosomes that don't function to break down lipids in the brains, so they start to accumulate, and the kid dies by the time they're 5. Cystic fibrosis, the mutation first started in Caucasians of European descent. 1 in 25 white people, Caucasians, is a carrier. So, 2 people that are carriers have kids, there's a 25% chance they'll have the disease. Now, the normal allele codes for the membrane channel that transports chlorine across the cell membrane. And we're going to talk about that now, but the kid dies by the time they're 5. Unless they get treatment, they can live into their 20s. Now, this is how it works. This is the protein that's made by the normal allele. If you have the defective recessive allele, 2 of them, then you don't make this protein channel. That's step 1. The second step is that you don't pump the salt out of the cells in your lungs to the space inside your lungs. If you don't pump that salt out, the water won't go from hypotonic to hypertonic by osmosis. If the water doesn't move from high to low concentration and makes the mucus in your lungs more liquidy, then the mucus starts to build up in your lungs, and you basically choke on your own mucus, your snot. So that's the real problem with cystic fibrosis. We're going to review how cystic fibrosis works. All right, codominance. Remember that in basic genetics, dominant allele is expressed over a recessive allele, and if you have the recessive allele, you don't have the phenotype. However, in codominance, both alleles are dominant and expressed fully and distinctly in a heterozygote. Here we have the black and white feather allele in chickens, and the chicken will have both the black and white feathers expressed equally, because black and white feathers are codominant in chickens. Now, don't confuse this with incomplete dominance. Incomplete dominance is a blending, like paint. So if feathers were incomplete dominance, then you'd see gray feathers, but that's not the case. Codominance is a little bit different. Sickle cell anemia is an example of codominance. If you have the big S, let's say that's normal allele, and little s for the sickle allele for hemoglobin, what ends up happening is you make both red blood cells that are normal and red blood cells that are sickle shaped. So, the whole problem with sickle cell is you get an adenine instead of a thymine. You get the wrong amino acid in the protein. Misfolded protein causes cells to sickle. It's pleiotropic, has a whole bunch of other effects. This ends part 5 of your genetics notes.